Hi there. Hello, Gail. Hello to my board members that are here, professional advisory board members, and volunteers and family members. Thank you all for coming today. My name is Nancy Udelson, and I'm the Executive Director of the Cleveland Area Chapter of the Alzheimer's Association. It's my pleasure to welcome you to our annual Foley Lecture today. This year's topic is one that we're really excited about, and as you all saw in your postcards, it's Searching for a Cure, a Conversation with Cleveland Scientists. We thought it was time to use our local experts. And with over 5 million Americans with Alzheimer's disease and no cure at this time, research is more important than ever. This afternoon, we will hear about some of the exciting Alzheimer's research being conducted right here in Cleveland. Cleveland is fortunate to be home to scientists who are leaders in this field, and we are happy to have 4 of them with us today. A big thanks to Dr. Brian Appleby, Dr. Gary Landreth, Dr. Bruce Lamb, and Dr. James Leverins. Before we get started, so that Tricia doesn't yell at me, I would like to draw your attention to the packet of information in front of you, which includes slides from the PowerPoint presentation you will view tonight. In addition, there is a note card to sign up for Trial Match and a brochure of the free services available through the Alzheimer's Association. Finally, an evaluation that we ask you to complete at the end of the program. To begin, we will view an eye-opening video created by Jennifer Curbo, a biomedical art student at the Cleveland Institute of Art. Jennifer developed this animation titled Involved in ALZ in support of Alzheimer's disease clinical research. The video illustrates what is going on inside of the brain of a person with Alzheimer's. We will also learn more about taking steps to help change the future of this disease. Dr. Brian Appleby is Associate Professor of Neurology at Case Western Reserve University School of Medicine. He sees patients at the Brain Health and Memory Center of University Hospitals. Dr. Appleby received his MD from Georgetown University and psychiatry and geriatric psychiatry training at the Johns Hopkins Hospital. His clinical and research interests include the diagnosis and management of human prion disease and young-onset dementias. Dr. Appleby.
Thank you, Nancy, and thank you for inviting me to talk. One of the benefits of being in our field is not only dealing with the patients and the family members, but also dealing with the advocacy groups and the support groups and the people that work in them. You really meet some remarkable people. This morning before I left, I told my wife not to wait for me for dinner because I was going to help out with the Foley lecture. And she said, what are you talking about, urine now? And those of you in the medical field will get that. We're not going to talk about urine, but hopefully I'll help set the stage for the rest of the speakers. I'm just going to give a brief overview about some of the research and how it applies to patients and families. So when we talk about research in any field, we talk about a really broad array of possible studies that people can do. So there's basic science research, which is done in a lab, looks at cells, maybe looks at mice or hamsters affected with the disease. And then from there, possibly people could just look at your disease itself, what's so-called a natural history study, because it's very important to know the course of the disease, especially such a chronic and long-term disease as Alzheimer's, to know whether or not different treatments are actually affecting the natural history of it. And then finally, we need to be able to diagnose these diseases accurately. So a lot of studies will look at diagnoses. And then finally, what many people are interested in, we have treatment studies. Treatments may look at specific molecules or drugs, or they may be non-pharmacological. They may look at behavioral interventions or even devices that are designed to improve symptoms or halt the progression of the disease. Mainly these slides are for your information. I'm going to really go through them so everyone else can talk. But there are a couple of things to keep in mind if you're interested in research. One is you're going to be asked to do a couple of things usually. They'll probably want to look at your medical records. It's very important that they make sure that you have the right diagnosis for the study. So they'll want to know a lot about you, and they'll ask you a lot of details. They may also want to talk to a loved one of yours or a family member, because a lot of times you have to get other information that the patient affected with the disease may not know or might not even realize is going on. They'll also possibly want to administer a paper and pencil test to see how your memory is. And they may also want to do some blood work or a brain scan to look at different things. And then finally, if you're in a treatment study, you may be asked to either take a medication or a placebo or a sugar pill. So what do you need to know when you look for studies? And this is an example for Trials Match or another type of research database that you're looking for. You want to make sure that you know what your diagnosis is, because all studies are going to be very specific about diagnoses. So, for example, there are some studies that will be specific to Alzheimer's disease, some that may be specific for frontotemporal dementia. So you really want to ask your doctor and get a firm grasp of what the diagnosis is. You also want to know what medications you're taking. Some medications may exclude you from participating in studies. Ironically enough, some studies may require you to already be on some medications. You want to know your other medical conditions, because that may exclude you from participating in some studies. And then you want to know what's required of you. Obviously, if you live in Baltimore and you want to participate in a study in Cleveland, it's probably not a good idea to sign up for something that's requiring you to come back every week. So you want to know how frequently you have to come back. And then finally, if you're in a treatment study where they give you a medication for your illness, a lot of times these are placebo-controlled. So it means either you have the chance of getting the active drug or the sugar pill, and you don't get a choice. That gets randomized. But sometimes after the study's over, they'll give you a choice of taking the active medication. So it might be helpful to actually ask that in the beginning. When you talk about clinical trials, you'll hear about different stages from phase 1 to phase 4. And ironically, this is kind of how our talks are going. I don't know if we planned it that way or not. But you'll have the preclinical studies. These are studies in the lab looking at the way cells react to different things, which Bruce will be talking about. Phase 1 studies are actually looking at whether or not a medication is harmful to a person. So they want to make sure that you're not having dangerous side effects. Phase 2 treatments are partly looking at that, but they're partly looking at what effect the medication is having on the person in a positive aspect. But they want to make sure that they're getting the right dose of the medication. And then finally, phase 3 is when people know that it's a safe drug and that they think they have the right dosage. They want to know if it's actually affecting the disease the way we want it to. So for Alzheimer's disease, is their memory stabilizing? Is it improving? Or does it continue to get worse as we would expect? Usually after that, if there's good findings and if it's a safe drug, the FDA will approve it. And it will be followed up by phase 4 study, which looks at a larger population, possibly for other indications or for other doses. So there are a lot of different ways to find out about research studies. You can always ask your doctor or Alzheimer's Association. There's several databases that you can look in. The Trials Match database in the Alzheimer's disease is excellent. There's also one called clinicaltrials.gov. And then you have 2 research facilities here in Cleveland that you can also take advantage of. These are just some of the studies that we're doing at UH, from frontal temporal dementia to Alzheimer's to asymptomatic Alzheimer's disease to prion disease. And that's it for me, and I think now I'm supposed to introduce Dr. Bruce Lamb. He is a staff scientist in the Department of Neurosciences in the Lerner Research Institute at the Cleveland Clinic. Dr. Lamb received his bachelor's degree from Swarthmore College and his PhD from the University of Pennsylvania, prior to a postdoc fellowship at Johns Hopkins University. In 1996, Dr. Lamb was recruited to Case Western Reserve University, where he rose from assistant to associate professor and finally moved to the Cleveland Clinic in 2005. His laboratory works on the basic science of Alzheimer's disease. In addition, Dr. Lamb is actively involved in advocacy for increased research funding for the disease. Dr. Lamb has received numerous awards and honors, including the Jennifer B. Langston Award from the Cleveland chapter of the Alzheimer's Association and the National Civic Award and Zavin Hictarian Lifetime Achievement Award from the National Alzheimer's Association. Here's Dr. Bruce Lamb.
Thank you, Brian, very much for the introduction. It's really great to be here and to see such a great crowd. I also want to reiterate, I think, what a great organization the Alzheimer's Association is in terms of, again, getting people educated as well as advocacy at all levels. But what I want to do today is tell you about, I think, a really exciting project that's here in Cleveland that's supported by the Alzheimer's Association as well as some private donors. And what this project is what's called a multi-center program grant, which is what it means is that there's several different scientists here in Cleveland, and we're all working together with a common purpose. And so the title of our project, which may sound like a lot of gobbledygook at this point, but hopefully it won't by the end, is the role of mononuclear phagocytes in Alzheimer's disease. And this, again, involves 4 researchers, myself at the Cleveland Clinic, Dr. Gary Landreth, who's here, who will be talking next from Case Western Reserve University, Dr. Sanjay Pimplekar, who's also at the Cleveland Clinic, and then Dr. Richard Ransohoff, who's also at the Cleveland Clinic. So I wanted to come back to this, sort of the video that you saw just a few minutes ago. I think is a really great demonstration of sort of what the current thinking is about the disease. And so what do we know at this point that causes the disease? So we would say that Alzheimer's disease develops when multiple genetic, lifestyle, and environmental factors work together to cause the disease process to start. Some of these genetic factors, I'd sort of say we're beginning to identify. We're still, I think, not quite where we'd like to be able to be in terms of being able to help people predict where you're going to develop the disease, but I think we're certainly better off than we were just a few years ago. Major risk factors for Alzheimer's disease include age, which is certainly the #1 risk factor for Alzheimer's disease, far and away. Family history is probably second to age in terms of importance as a risk factor. And then there are some environmental factors. Head trauma, which is one probably you've heard a lot about in the news recently, both in returning veterans as well as in football players and hockey players. There's now increasing evidence that head traumas and even repeated head traumas are probably an important risk factor in terms of Alzheimer's disease. And then we also know, though, that individuals who have been on long-term non-steroidal anti-inflammatory drugs, so this is like the ibuprofen that a lot of you probably already take, there's some evidence that people who are on these for a very long period of time have reduced rates of developing Alzheimer's disease. But importantly, there's only thus far only 5 FDA-approved drugs for Alzheimer's disease, and probably I don't need to tell this audience that all of these really only treat the symptoms and really don't halt disease progression. So obviously we need new and better treatments which really attack the disease and stop it. And I would like to sort of say, too, that therapies that target amyloid, which was this protein which he said develops in the trash cans and overwhelms the cell, that there are now a whole variety of different therapies that are being developed to target amyloid. But all of those that have reached this phase 3 that Dr. Appleby was talking about have all failed thus far. And so while we have certainly not given up hope on amyloid therapies, and there's continuing to be even more trials and even more drugs, and you'll hear about one of them in just a minute, I think it's also worth thinking about that we need new investments to think of new ideas and new targets for Alzheimer's disease, as opposed to, again, just those ones that were mentioned there, this amyloid protein which accumulates in those trash cans, and then also the tau protein. So that's part of what this project was, this collaborative project between the 4 of us to target sort of new pathways and identify new targets that we can then develop into therapies in the later stages of this. So that's what we really need is new therapeutic targets and to bring these into clinical trials. So what is the target that we were interested in? Well, so in addition to the neurons that you saw in that video, which are the major cell for communication and how memories are formed, how our thought processes occur, there are other cell types within the brain. And one of them that we've now begun to appreciate is far more important, I think, than previously appreciated, are immune cells. So there's an immune cell of the brain, and it's called a microglia. And it's shown, this is a picture of a bunch of microglia here, and this is at its core, this picture here is an amyloid plaque. So what these innate immune cells, these microglial cells, do is they come in and surround the amyloid plaque and essentially try to remove it from the brain. That's their normal job, actually, within the brain. For some reason, and this is something we still don't really understand, at some point they fail and can no longer remove the amyloid, and it starts to build up within the brain. But in addition to these microglia, there's now increasing evidence that in addition to these immune cells, there's also, looks like, movement of peripheral immune cells into the brain. Typically, the brain is thought as to be this very, the only immune cell typically within the brain is the microglia. But in certain disease conditions, and we are now beginning to think that Alzheimer's disease may be one of them, there's also some evidence that peripheral immune cells can enter the brain. And so this is another target that we're interested in. And then finally, and I think this is most importantly, there's been recent genetic studies that identified some new Alzheimer's disease genes. And a lot of these genes are only expressed in these innate immune cells, the microglia and the monocytes. So it suggests that whatever is going on within those cells actually is directly linked to the disease. So that's why we really were interested in now studying these processes and these cell types within the disease to see if we could identify new targets for the disease. So we do this using animal models. As Dr. Appleby mentioned, we can introduce familial Alzheimer's disease genes into mice and then study how the brain pathology develops, how these neurons that you talked about, how they function. And then we can also even examine behavior, learning and memory within the mice. And I know that I'll probably get questions later on of how we analyze mouse behavior, but we can get to that in the discussion or the panel section. But our idea here is that, again, we have these resident microglial cells, and then you also have these monocytes, which typically are normally only in the blood vessels and never enter the brain. At least they shouldn't normally. But in disease, we get both these tangles that accumulate within the neurons, as you already saw, and then we also get these beta amyloid plaques. And so the real question is, we get these two different cell types now coming within the brain, And so 2 of the projects were really interested in how these cell types interact with amyloid and potentially try to remove it. And are there things we can do to promote its removal? And then the second 2 projects were really focused now on how these cell types interact with this neuron, which is aggregating tau within the cell. And so between these 4 projects, we were really hoping to now begin to understand what role these different cell types have in disease. So what are our results thus far? We're about halfway through the project. It was initially a 3-year project. So one of the things that we've really become interested in and is directly relevant to this project is this new Alzheimer's disease gene, which is called TREM2. This is one of the ones I mentioned earlier. And it's called TREM2 because it's short for triggering receptor expressed on myeloid cells 2. So if you can say that several times fast. But what the idea is, basically, this is a gene, again, that's only expressed within these microglia and monocytes. And typically what it does is it sits on the surface of these cells and is actually a way for it to interact with its environment. And so when we looked in the animal models for Alzheimer's disease, we see that these TREM2 positive cells are accumulating around these amyloid plaques, just like I showed you before. So what happens is TREM2 is upregulated as these cells start to accumulate around these plaques. So I think TREM2 represents, I think, a potential very important new avenue and potential therapeutic target, actually, in disease. And that's something we're currently studying very hard. And the other, and this shows work from Dr. Landreth's lab on his project. We've also been interested in blocking inflammation in the Alzheimer's disease models. And this is one study where they used a genetic alteration in these microglial cells to alter how they respond to amyloid. And when doing this, we could actually get enhanced sort of removal of amyloid from the brain. So this is another, again, potential target within microglia itself, a signaling pathway that we think will be important to further analyze and determine whether we could target this therapeutically. So the projects, again, there's 4 of them, as I already mentioned. The first one is by Dr. Ransohoff, the second one by Dr. Landreth. Again, I'm not going to give you, I have the names up here, but I'm not going to go through them because I just don't think it'll be terribly helpful. And then the final 2 projects are both by myself and Dr. Pimplicar. The long-term goals are really to get insights into the role of these 2 cell types, again, these monocytes and these microglia in Alzheimer's disease. Again, this is an area that is relatively thus far unstudied. And so we think that this will provide novel insights, and we already have some of them already, I would suggest, into the roles of these cells in disease pathologies. And most importantly, it will help us identify new potential therapeutic targets, and that's really what we want, where we want to be. And in future studies, we'll obviously have to now start to think about, okay, now how do we target this in doing preclinical studies, targeting those therapies in our animal models to see if they're effective, obviously before they can then eventually go on to human studies. So that's why this process is very long and unfortunately sometimes frustratingly long for both us and I'm sure for all of you. But what's, I think, important to note out is this multi-center program project is really the first and only of these types of grants that the Alzheimer's Association has ever funded. So it's really unique that this is here in Cleveland. And it's a unique collaborative interdisciplinary study that's based here in Cleveland. And it's also a partnership between researchers at both of the 2 medical institutions in town. And what's also interesting about this program project, this would not have been possible without some substantial financial commitments. And really this is sort of another way that this project is very unique, that it's a partner between the Alzheimer's Association and then also private philanthropy. And in particular, Jane and Lee Seidman, who are really able to come in and help support this project. Otherwise, it would not have been funded. So that's really sort of been unique. And in here, I'm going to put on my advocacy hat just for 1 second. That this type of support is really important to move these studies forward. We would not have been able to do this if we hadn't gotten the funding to do it. And funding right now through National Institutes of Health has never been tighter. The sequester cut that happened earlier this year cut most of our budgets by about 10%, right off the top. The cut that's supposed to take place in January, unless, again, there's a resolution to the budget, it's estimated to be about 25% on top of that. So, again, this is something I think everybody should be aware of and advocating it every way you can possibly think of because it's just too important. So these are the folks. This is Dr. Pimplicar. This is Dr. Ransohoff, and of course this is Dr. Landreth right here. So with that, thank you. With that, I'll now introduce the next speaker. And this was intentional to sort of go from the basic science through different stages of clinical trials. So the next stage is really now thinking about, okay, now we've identified a therapeutic target, where do we go from there? And so with that, I'd like to introduce Dr. Landreth. Dr. Landreth is a professor of neurosciences at Case Western Reserve University. He has worked for the past 25 years on Alzheimer's disease at CWRU, investigating the basic biology of the disease. More recently, his laboratory has been working to develop new drugs to treat Alzheimer's disease, and I'm sure some of you have heard about his studies on bexarotene, which was been a lot in both the local and national press, and I think that's what he's going to be telling us today.
Good afternoon. So I want to thank the Alzheimer's Association for inviting me to be here this afternoon with you. And the other thing is the support of the association has been absolutely critical for the work in my lab for the last 10 years. I can't tell you how much that money means. It's really valuable, and I thank you for your support of the organization. So what I want to do is to tell you a bit about some work we've had been going on in the lab for about 15 years, which is actually only recently sort of borne fruit. So as the introductory talks discussed, this is the disease of the 21st century. It is, and we are in the midst of an epidemic. There are no drugs to treat this disease. Then there's urgency in generating new therapeutics. Now, over about a decade, we came to understand aspects of the disease. And so one of the things which was mentioned earlier is, as you're listening to me, we're all producing prodigious amounts of amyloid just as a normal consequence of the electrical activity in your brain. Now, when you're young and healthy, this amyloid normally gets cleared out and flushed out of the brain, and you maintain your cognitive abilities. Now, it's like everything else. As you age, things go awry. And one of the things that happens is that these systems to clear amyloid from the brain slowly become less efficient and decay. And this happens to greater or lesser extents at different times with different people. So some people will exhibit these cognitive changes associated with the failure to clear these amyloid species. And some of us may never get the disease because these systems remain intact. Now, the key thing here is that these small amyloid species are what perturb the ability of one neuron to talk to the other. And as you accumulate the amyloid species in the brain, you experience failures of memory and cognitive defects. We believe that this is what underlies the primary symptom of the disease, which is loss of memory. Eventually, these amyloid species accumulate, they stick together, and they form the amyloid plaques, and that's typically been thought to be representative of advanced stages of the disease. So, now, that being true, our contribution to this discussion arises from an observation we made several years ago that we've basically figured out what the garbage disposal was. And it's a molecule called apolipoprotein E, or ApoE. And what this molecule does is it facilitates the clearance of these soluble beta species and having them degraded. So it follows then that if we could make this process run more efficiently, we should stay smarter longer, right? So one of the things that's happened in the fairly recent past was the recognition that we are able to elevate the levels of ApoE in the brain. That is, we're enlarging the size of the garbage disposal, if you will. As one of the drugs that does this is a drug called bexarotene, which we discovered about 4 or 5 years ago. And this is a fantastic drug because what it does is it elevates ApoE in the brain. It's FDA approved, it's orally delivered, and it has a really great safety profile. So we moved these studies into mouse models of Alzheimer's disease with the idea is that we're going to give this drug to a mouse. They're going to elevate their brain ApoE. We're going to drive this clearance process more efficiently, and the mice should stay smarter longer. That was the idea. So What we've done now, and work we published about a year ago, was that we were able to take mouse models of Alzheimer's disease, give them this drug, and within just a matter of a few days, we were able to improve their behavior because what we were doing was we were more efficiently clearing these small amyloid species out of the brain. So, let's be quite clear about this. These are mice, and not get ahead of ourselves. It is imperative, because truth be told, mice have not been very good at predicting what drugs are going to work in human beings. So the question then is, does this drug work in human beings in the same way it works in a mouse? So what we've done is we've initiated a small clinical trial together with a company called C2N Diagnostics in St. Louis, which has this really fancy technology. And so what we're about to embark on is a study where we'll take normal patients, probably college kids, bring them into the clinic and administer the drug, and then using a really sophisticated technology, use isotopes to tell us how fast ApoE is being generated in the brain and whether that affects and accelerates the clearance of amyloid from the brain. Now, so one of the interesting features about this is extraordinarily expensive. It's about $100,000 a head for these patients. But the good thing is it takes rather small numbers of patients. We're going to use 6 patients on placebo, 6 patients on drug, and we should be able to tell whether the drug works in humans as it does in mice. So we expect to have our first subject in the clinic in the next month. It's just taking longer than we thought. It'd be 10 weeks from start to finish. We want to have all this stuff collected by the first of the year. It remains to be seen, but we're actually, this is what's been called a biomarker study. But it is an absolute prerequisite to going forward into larger studies, which are going to cost many millions of dollars. And so we're going to get a fast read on these biomarkers over relatively short periods of time. So what we have now is this is our phase 1 trial, which is underway. And I hope to tell you what the outcome is after the first of the year. In parallel, there is an important study that's being carried out by the Cleveland Clinic in Las Vegas at the Lou Ruvo Center, which is termed a phase 2 trial. Now, what they're doing in Las Vegas is they're enrolling individuals with diagnosed Alzheimer's disease, putting them on drug for 8 weeks and asking the question, is their memory improved on the drug, and is amyloid being cleared out of their brain? So that study is, I'm told, about halfway done, so the outcomes of that should be known in the relatively near term. So with that, I would like to get a great pleasure to introduce Joe Leverance, who's about to arrive in Cleveland. Dr. Leverance is currently professor in the Department of Neurology and Psychiatry and Behavioral Science at the University of Washington, Seattle, and he's a neurologist in the VA Puget Sound healthcare system. In Seattle, he's been an investigator in the Alzheimer's Disease Research Center for over 15 years, and more recently has also directed the clinical research programs for Pacific Northwest Udall Center for Excellence in Parkinson's Disease. And the really cool thing is he's coming to Cleveland in January to run the Lou Ruvo Center in Cleveland. He will be the Joseph Hahn Chair of the Cleveland Clinic Neurological Institute. And Jim, we're delighted to have you.
Thank you, Gary. Great introduction, and thank Nancy and the Alzheimer's Association for inviting me. As was stated, I'm going to be new to Cleveland in January, and I'm looking very much forward to it. A lot of exciting opportunities, as you already have heard about, and hoping I can add my 2 cents worth into the program here. So my job in the next 12 minutes, is that what I have? Is something like that? Is to talk about what biomarkers are really, and actually you've already heard a lot about biomarkers already in terms of what Gary was talking about. In terms of some of the outcomes of his study, we'll be looking at biomarkers, which gives us a much better idea of what's going on with the disease. You've already kind of heard what Alzheimer's disease is, so I'm going to sprint through this. But I do like always throw a little bit of history in it. It was really Alois Alzheimer that originally described the pathology that we still really hang our hat on for Alzheimer's disease. So just to quote from some of his writing, he talked about the brain atrophying in Alzheimer's disease, and that he saw in the brain cells these tangled bundles of fibrils, which are those tau-containing proteins that aggregate in the cells. And then he talked about that there are these sites of deposition of a peculiar substance, which is the amyloid. So right from the beginning, he recognized the major pathologies of Alzheimer's disease, of what we now call Alzheimer's. So this is a kind of a typical stain we use to look at diagnose Alzheimer's disease. And what you can see are these amyloid plaques here. And within the brain cells, you can see this dark areas, which are the neurofibrillary tangles. And let me see what I got here. And in these plaques, if we go really hone in on them under the microscope, you can see in the center is the amyloid, or what we often call the beta amyloid protein, or BAP sometimes. And around these plaques, as mentioned earlier, we often see a lot of inflammatory cells, which is why there's such an interest in how inflammation plays a role in the disease. The other pathology, actually, again, which Alzheimer described, and here are some of his drawings, are these tangles in the brain cells. And you can see these are cells just packed full. And you can imagine, while I think most of us agree that it seems as if it's the amyloid that gets the pathology going and gets the disease running, that it's actually the tangles in the cells themselves that correlates with the dementia, with the dysfunction. And that's very important, because ultimately, if the amyloid therapies don't work, we may need to look at how tau therapies work. Because if you have a bunch of amyloid in your brain but you're thinking okay, then who really cares, right? And I always throw this in. This is the genetics of tau, which is these tangles, and I always like to say you'll be tested on this later. So what is a biomarker? And if I always have a chance to quote Yogi Berra, I always like to. So he says here, you've got to be very careful if you don't know where you're going, because you might not get there. And I think that's very much what a biomarker is. A biomarker is telling us what's going on with the disease, what are our drugs doing with the disease, is it actually affecting the disease itself? Because our best biomarker right now is cognitive testing, and it often takes 6 months, a year, or longer to really figure out if you're doing anything with the disease. If we had a better biomarker, say a blood test, a spinal fluid test, an imaging test, that could tell us how the disease is going on a week-to-week or month-to-month basis, we can much more quickly decide, is a drug really working well for the disease? So this is a quote from one of the dictionaries I looked up online. It's a substance to use as an indicator of biologic state, and it can be used in a number of different ways. Obviously, one thing is it can improve diagnostic accuracy. So do you have Alzheimer's? Do you have frontotemporal dementia? An area, as was mentioned earlier, that I work in is Parkinson's dementia, where about a third of patients have coexistent Alzheimer's disease. And in that example, we may want to know which one of them should get Alzheimer's therapies and which ones should not. This is where a biomarker would be really helpful. It can monitor treatment response. Again, hopefully a biomarker can tell us if we're getting a response in a month or two rather than waiting 6 months, 12 months, 24 months. It can guide best treatment. Anybody familiar with breast cancer knows not all breast cancer is treated the same. And I think we're increasingly finding that biomarkers are telling us something about Alzheimer's, that not all Alzheimer's is the same, and that we may want to treat different kinds of Alzheimer's differently. It can actually also predict dementia, and people say, well, why would we want to know that? But the day we have a preventative therapy is the day we want to be able to predict it. And sometimes even biomarkers tell us something about the disease we didn't already know. Potential biomarkers, so you said we weren't going to talk about urine, but we will talk about urine here just to mention it. So biomarkers can be pretty much anything, any fluid. I hate to say it, but even in our Parkinson's work in Seattle, we have what we call a poop for PD study. So Parkinson's study looking at stool. So we'll look at anything. And blood, urine, spinal fluid. We're finding that the spinal fluid often is the best site of biomarkers because it's really not adulterated by things from the rest of the body. It really tells us what the chemistry of the brain is in a person who's still alive. We don't have to wait for the autopsy, so to speak. Neuroimaging is something there's a lot of excitement about, amyloid imaging in particular, and being able to image those plaques in the brain. And there's some really very recent imaging studies that are starting to suggest that they can see those tau aggregates in the brain cells as well. So this is an area that's rapidly evolving. But there's no doubt that this is where the future of modern Alzheimer treatment is going to be. We're really going to need these biomarkers to tell us how best to treat people, if treatments are working, etc. This is just a little bit of data just to show you. This is showing Alzheimer's disease and that amyloid concentrations in the spinal fluid. And what you can see is that in a normal control, there's sort of this upper level here, let's say about 200 on average, in a normal control in the spinal fluid. In mild cognitive impairment, the sort of pre-dementia state, it's starting to drift down, and then it's way down below this 200 mark in people with Alzheimer's disease. And in fact, you look at this data, and you are starting to get to the point where you can start to identify people who are at high likelihood to develop Alzheimer's. I think, Brian, you were mentioning the A4 study, which I believe will be trying to identify these people who are at high risk but don't yet have Alzheimer's, trying to intervene even before you develop your symptoms. We also find, interestingly, in Alzheimer's, those amyloid levels in the spinal fluid don't go down, don't go up, they go down. And the thought is that amyloid is bound up into the plaques in the brain, and so it's not released into the spinal fluid. On the other hand, as cells die, they release that tau protein that's in the tangles, and we can see in Alzheimer's disease that tau levels are actually elevated compared to people who are normal controls. And actually, the greatest level of tau we see increase really is in Creutzfeldt-Jakob disease and prion disease, where there's massive cell loss. We see these very high levels of tau in the brain. And currently, we can actually use the combination of that amyloid and tau level in the spinal fluid as a diagnostic test for Alzheimer's disease. I think generally we don't use that commonly in the clinic. We do on cases, I think, where we're uncertain what exactly is going on, and you actually compare what you're looking for basically is elevated tau levels but low amyloid. So down in here are where we're going to see people who likely have Alzheimer's or pre-Alzheimer's disease. So does this sort of tell us anything? And as I said, there's a bit of a predictive value. These are one study coming actually out of our group in Seattle showing that individuals who had abnormal spinal fluid, so low A beta, high tau, but were normal, were more likely to decline cognitively than people who had normal spinal fluids at the beginning of the study. So suggesting that we actually may be able to predict who's at higher risk for developing Alzheimer's disease. We have a number of studies that have been going on trying to modify amyloid, and this is one study we've been doing in Seattle looking at how statins, drugs are usually used to lower your lipid levels, how they might actually, in fact, influence amyloid and tau in the brain. And so, again, as a preventative therapy. But this is a way where we're interacting biomarkers with a clinical study. I think I'm actually doing pretty well here time-wise. I'm very proud of myself. And this is just some data out of this showing that simvastatin we're showing actually lowers tau levels, so goes in the opposite direction of where we would expect for Alzheimer's disease. So we're starting to use these biomarkers to tell us what drugs are going to be best in terms of treating Alzheimer's disease. This is another study, resveratrol, many of you heard about. This is the wine extract. Unfortunately, you have to drink, I don't remember what it is, something like 40 or 50 bottles of wine a day to get the amount of resveratrol. I do like a glass or two at dinner at night, but I'm not quite up to that level. So, and this study's been ongoing, I think. Are you doing it at the university? I can't remember if Alan, are you doing that study? Yes. I think that's fully enrolled, so we should hopefully have some data on that, and not too distant future. Yeah, thank you. And that's it. So thank you very much. Okay. So back to you, Brian. And
I have to say, I'm very impressed. Everyone kept on time, which is saying something. At least I know it is for me. So now is probably the most important part of the night, and that is question and answers. This is really your opportunity to ask anyone on the panel here any question that you have regarding neurodegenerative illness, Alzheimer's disease, Parkinson's, frontotemporal dementia, and we'll take any questions if anyone has any. If you all coordinate the way you raise your hand, she'll go in a circle.
Hi, Ray Antonelli. The two Phase 1 and Phase 2 tests at the Lou Rubulo Institute in Las Vegas, those appear to be going on simultaneously when you said initially that Phase 1 comes first, Phase 2, then Phase 3, et cetera. How did that happen, and what's going on there?
So that's a good question. So these are two independent efforts, and truth be told, it's basically about the economics. We have taken a conservative approach. We raised $1.4 million to run the small $1.4 million to run a small clinical trial. And just because we're basically conservative, I think many of these big Phase 3 trials have failed because the people who ran them and the pharmaceutical companies didn't go back and verify the drug did in humans what they thought it would do. So we thought we would start off and validate that the drug did what we thought it did in humans. The Cleveland Clinic Lou Ruvo Center had a donor who came in and provided dollars to them to directly test the disease in Alzheimer's patients, and that's basically what's allowed that trial to go forward. I think it's an interesting parallel. We'll see how this turns out. We're terribly excited about this.
The FDA approval.
The other piece of this is an FDA-approved drug. So there are no regulatory barriers going ahead with trials. And moreover, there really is no regulatory barriers, and people are taking this drug off-label, which we do not recommend.
What was it approved for originally? Something else?
This was originally approved for T-cell lymphoma. It's a skin cancer. It's not a very good skin cancer drug. It's not very highly prescribed, but it has the virtue of being a nice, safe drug.
Gary, could you talk about some of the reasons why people shouldn't take it off label?
Okay, so let's be clear about this. We've tested this drug in Alzheimer's mice, and the mice have been poor predictors of success in humans. So I think we have never tested this drug. No one's ever tested this drug on a diseased brain, individual with a diseased brain. We don't know what's going to happen, and I think we need to take a very conservative, methodical approach to trying these drugs in people with Alzheimer's disease. So.
If I could just add to that, I think there's another point too. Obviously, the safety point, but also the scientific point too. We had this problem when we looked at Namenda and frontotemporal dementia. You can get Namenda; it's approved for Alzheimer's disease, and it was really hard to study that disease in FTD because everyone was taking it off label. So we didn't have nearly the amount of participants in that study that we thought we would initially, and it was hard to find people. So it's harder to actually find a drug that works if people aren't, undergoing the scientific method that tells us whether or not it's a useful drug or not. And that's aside from the safety concern. Okay.
So I'm Paul Ford. I'm a medical ethicist at the Cleveland Clinic. I just wanted to follow up with a point that Dr. Lamb made. Although this isn't under FDA regulation for research, both of the institutions, UH and Cleveland Clinic, have very strong institutional review boards that do oversight. So these aren't maverick researchers who are just doing it and don't need regulation. And you're welcome to say more about this, but I just want to make sure everybody recognizes these are still have oversight and are being done properly and well.
right? I mean, I think we all agree on that, and I mean that was certain. I'm glad you clarified that. All of these go through a rigorous any study that you're going to put out there, and obviously doing treatments in people have to go through a rigorous program of institutional review. So yes, absolutely. And ongoing oversight as well, right? Continuing to sort of monitor that study as well.
I have a question pertaining to the drug companies and amend and a few things that were brought up. How does that happen, that billions and billions of dollars are spent on these drugs since 1996 with really no proven efficacy? Insurance companies pay for them. They don't pay for many medications for different. How does something like that happen? Is that upsetting that these kinds of dollars are spent in our society on drugs that don't work, basically?
Are you talking about the approval process itself or after the drugs have been approved? Yeah. Well, drug companies are companies, right? So the question was, how do drug companies spend millions of dollars and not have these useful drugs, and how do they justify that? And how do they get licensed to do that? I think what you're finding is that more and more drug companies, because it is a business, are getting out of the business of neurodegenerative illnesses, because the amount of money that goes into research and development of trying to find a biomarker or a potential hit of a drug, and then mouse models and clinical trials is many, many, millions and billions of dollars. And very few of them even make it, under 1% make it. Does that answer your question?
I thought you were referring to the currently approved drugs.
Yes, and how they, number one, became approved, and when you say about other drugs, if in fact there were an antibiotic for gram-positive diseases that became to market and it didn't help, I think someone would stop it. And this doesn't seem to stop it.
Well, in order for the FDA to approve a drug, it has to be safe, but it also has to be able to affect a certain endpoint. So when people look at medications for Alzheimer's disease, there are various different endpoints people can look at. You can look at how their cognition is. You can look at how their level of functioning is. You have to think the medications we have for Alzheimer's now isn't your typical medication, right? If you have a pain and you take a pain pill, you expect your pain to get better. If you have high blood pressure, you take a high blood pressure medication, you expect the numbers to go down. That's not how these drugs work. It just slows the decline. And they've been able to show that in studies, and that's how they got approved.
Good afternoon. You mentioned in one of your slides ibuprofen, those on nonsteroidals, is reducing the risk of Alzheimer's disease. Has there been any study, with those who have Alzheimer's where ibuprofen may slow the process or show any other kind of results?
There's actually been quite a lot of work done in that area. And unfortunately, what's happened is we do know ibuprofen has other side effects, including increasing cardiovascular disease outcomes, and that's what they saw in those studies. I don't know of any data off the top of my head, but there were some long-term follow-up of those initial studies to see if the people who were initially on a year or two of ibuprofen versus placebo came out better. And I think the data's been mixed, has been my interpretation.
There was a very big study called the Cache County study, which is a county in Utah.
Yeah.
And they followed these people, and they looked at ibuprofen usage. And the last paper I remember that came out to them is ibuprofen usage tended to actually affect people differently depending on how rapid their disease course was to begin with. So I think it goes back to your point about different types of disease.
So it still needs to be explored, I think, but much like Bexarotene, we're not recommending people go out and start taking ibuprofen at this point. And that's actually, I've seen a lot of people who've done that, but we don't. I think we're all pretty convinced that the inflammatory system is important in the disease. We just don't know exactly how to attack that issue yet.
And I think also in terms of what we've also heard a lot about, including Dr. Leveren's, even when we should target therapies. And I think that's sort of becoming an increasingly looked at thing with regards to all of the clinical trials that are going on Alzheimer's disease, because we've just now learned about the past decade a lot more about the disease because of these biomarkers. That now, for example, we appreciate that the disease probably starts a decade or maybe even 2 decades prior to sort of clinical manifestation. And so then sort of really the big question becomes, okay, when you're targeting therapies, when in disease course do you target, right? So you have to know sort of when those specific targets are engaged and when you're going to be able to successfully attack those targets. And I think that we still really don't know, especially for like inflammation or even for amyloid for that matter. We know amyloid occurs early, but when exactly during that process should we be intervening is still a big and active question in the field.
Hi. I just wanted to know, in your research, have you found that there have been large amounts of copper or iron in these amyloids or any part of the plaque that's on the brain? I had read that that was one of the things also that they found with this disease.
I have some information. I like to call, my old mentor used to call plaques the black holes of the brain. And a lot of things tend to get aggregated on them and included many minerals. I know zinc was associated with it. I can't remember about copper and iron. There was a recent iron study that came out. I can't remember the exact results of that. But there people have been looking at that. Do you have something, Gary, that...
So there's a hypothesis that indeed copper and iron are involved in pathogenesis of the disease. There's a company called Prana who had a molecule which would take copper out of the brain. Those trials have so far failed. So it was a live hypothesis, probably not true.
Have there been any studies or anybody looking at people that eat certain things, like people that are on a vegan diet, because those have been associated with reducing inflammation? So just look maybe longitudinal studies of people that are on a vegan diet versus on a regular diet and whether or not they're less prone to Alzheimer's.
So the question was, does diet have any effect on Alzheimer's? Do you want to tackle that one, Jim? Maybe the Mediterranean diet?
Now that I have a microphone, there is some people are definitely looking at that. I'm not sure that we have the data yet to tell us for sure. I think one thing that's really important to recognize is that when we look at people who've passed away with Alzheimer's disease, they more often than not, especially if they're older, have multiple changes in their brain going on, not just Alzheimer's. For example, vascular disease is extremely common. There are more people with Alzheimer's who die who also have small strokes than people who die just with Alzheimer's alone. So people always ask me, what should I be doing? Well, not having those strokes is definitely a good thing. And we know for sure that having a good diet and exercise, all the things that are difficult that we've been hearing about for many years, are going to help you with that component. Whether they directly impact the Alzheimer's, I don't think we know yet. But people are looking at that. There's a cooperative study amongst the Alzheimer's centers that's going to be looking at exercise and Alzheimer's disease. So people are starting to look at things like diet change, fat content, exercise as modifiers.
These are difficult things to study. For example, people have noticed a lot of associations with a Mediterranean diet and decreased incidence of Alzheimer's. But is that the diet, or is that actually the community atmosphere that comes around their meals and the social and cognitive engagement they get from that? Who knows? It decreases the risk.
I kind of went to sleep during the part where you're talking about statins. Is it good for a statin to cross the blood-brain barrier, or is it bad for it?
So we think it's good.
So simvastatin is a good drug. I have a mild cognitive impairment or early Alzheimer's, depending upon which office I'm at. And I would want to take the cholesterol medicine that was better for my brain if I'm taking one because it's the fashionable thing to do.
Right.
So simvastatin would be fashionable right now for me.
Well, the statin studies right now we're just starting to look at how they affect the Alzheimer-related biomarkers. What statin studies have been done in Alzheimer's have not shown an effect on Alzheimer's so far, but we're not sure they picked the right statins, and we're just, again, this study that I showed you was in younger individuals, people in their middle ages, who...
I'm not that old.
Well, we may be asking you to come in and participate in one of our studies. But we're not recommending a specific statin at this point, but our hypothesis is that the statins that get into the brain are the ones that may be beneficial. But we don't know that yet. I don't recommend, again, like the ibuprofen, that you go out and start taking statins for your memory loss. There are several, and I wish I could name them all off the top of my head because there are a lot in this class, but there's some that do and some that don't. And that's where I think the literature has been a little complicated.
Jim, one question that we get a lot of times in the clinic is because of the black box warning that came out for statins that it could cause memory impairment, whether or not patients should be taking statins. What do you tell your patients?
My understanding of that side effect, that it's sort of a relatively what we call acute change, that some people will have an initial reaction that will cause confusion. But the one nice thing we have about, although they were negative, the statin studies in Alzheimer's, they didn't show a decline in cognition. They actually showed just no change. So generally speaking, I mean, I individualize it. Somebody comes in, they want a statin, they thought there was a big decrease, then we change the statin or we stop it.
I think it's important. You said it's not good to have strokes.
Right.
And statins help prevent strokes.
That's also not good for your cognition.
Right.
When I was a nurse, I went to a seminar at Johns Hopkins. It was on diabetes. And there was a doctor from Japan, and he said one of the newest things is ACE inhibitors that are able to cross the blood-brain barrier have slowed down dementia. That was like many years ago. Have you heard anything about that?
That's not ringing a bell for me. There was a recent study, I think, that came out that looked at different ACE inhibitors and decrease of dementia. These studies are hard because, as Jim stated, most people that have dementia don't just have one cause of dementia. It may be vascular disease, it may be Alzheimer's disease. So it's hard to know what these individual drugs are affecting. And you'll see some studies with the same medications that show benefit and some that don't show any benefit. So I think it really is still up in the air.
But,
Good afternoon. And I know that Bruce or Gary is dying to answer the question. So how do you know when mouses behave differently?
Do you want a demonstration?
Okay, so that's a really interesting question. And it, I think it's representative of kind of where we are. We're using rodents to test drugs on about human cognition. And so I mean, that's a little weird. Mice actually are reasonably intelligent animals, and we can test their ability to remember. We can test anxiety. We can test different aspects of cognition using rather specialized and kind of bizarre tasks. We ask them, for example, we throw them in a pool of water. We ask them to find a hidden platform. We ask them to find novel objects. We ask them to burrow and find Cheerios. And it's a black art. But this is basically what's available to us. I don't know, Bruce, you want to?
Yeah, I mean, I think you try to pick, I think, tasks that are both, so we'll hit some of the sort of the same, areas of the brain that we think are targeted in Alzheimer's disease. Also, sort of will be good assessments of that in these animal models. So some of the tests that Gary mentioned, this maze, this pool maze is one of those that hits the hippocampus, which is sort of one of the major places that Alzheimer's disease hits. There's others, object recognition, ability of the animals to actually recognize whether something's new or different, because a mouse will always go and investigate something that it's not seen before. So if you put in 3 objects and one is the same, or 2 are the same that it's seen before and one is new, it'll spend most of its time investigating that new object. So those are the kinds of things that you can do. They're relatively quick to do, which is sort of one of the important things for the research side. Is we want to be able to get a good, relatively quick way to assess whether their memory is intact or not. But I agree with Gary that it is at best imprecise in terms of really determining whether that's the same type of memory impairment that we're seeing in humans. I think we could sort of say that some of the same brain regions and pathways are impacted, but I don't think we can say definitively that that is exactly the same as what is seen in humans, for sure.
Could you talk a little bit about Alzheimer's disease models in mice and how the pathology compares to humans?
Yeah, I mean, it's also imprecise, although it's interesting because I remember as a graduate student, I had put in a grant to develop an animal model of Alzheimer's disease, and I was told that, mice don't live long enough. This was my fellowship that I had applied for, that mice don't live long enough to develop any of the Alzheimer's pathologies. This was sort of, which I carried around for me the rest of my career, obviously. And so, but at the time that was true. We didn't have any animal models. And then through sort of a lot of the very powerful genetic technologies, we were able to actually introduce genes into mice that, again, in humans cause Alzheimer's disease, and show that if we introduce these into mice, we can induce, again, some of the pathologies you saw, which, for example, the amyloid pathology is very well now replicated in multiple different models, multiple different ways. But, again, it's an imprecise model. In those models, we don't get all the rest of the pathology, so we don't necessarily get all the tau pathology that's seen in humans. There's other models that develop tau pathology, but obviously they don't develop all the amyloid. So again, that's where I think we can model certain parts of the disease very effectively, and I think very clearly. But again, I think the question comes back to, and that's why I think the important thing is to move these preclinical trials very quickly and try to get them into humans, because I think that's where we need to be, to really assess, what we're finding out in mice is really going to pan
out. My name is Sanjay Pimplikar. I work in Cleveland Clinic. I just had a couple of comments on questions that were asked on the vegan diet and also on the non-steroidal anti-inflammatories. And I think sitting on the dais, and when you're wearing a hat of scientist, you try to be very precise and give the wishy-washy answer, which may or may not be helpful for the audience. But what I would like to add on is, just last week a paper appeared in the Annals of Internal Medicine. Very good study. Like in the United States, we were always proposing potatoes and steak diet in the 70s. And so what this study did was, it looked at the people who had that diet from the 70s on, and then followed up what happens to them now in the 2000 and 2010. And it was not an Alzheimer's study. It looked at all the things such as diabetes, cardiovascular disease, and dementia. And in respect to what Brian was saying, that the Mediterranean diet may be helpful, but it may be the atmosphere or environment. I think we don't need to be so circumspect anymore. The data is coming out so clear. If you have plant-based diet, if you have exercise, those are the strongest protective factors. If you adopt them, they're not going to hurt you. They may or may not help you, but they will not hurt you. And so, it's not a single study on a Mediterranean diet. There are at least half a dozen or a dozen studies that they show that cognition is improved, or at least the decline is repressed. So I don't know why we as scientists cannot be a little bit more forceful in it's not a treatment, it's not a cure, but the data is coming out stronger and stronger. Lifestyle changes, plant-based diet, exercise, social contacts. That's the only thing that seems to be working so far.
I was looking for information relating sleep apnea to Alzheimer's disease. I was asking questions about this because my husband seemed to change so much on the days when he wore the mask a long time compared to when he didn't wear it at all. He has Alzheimer's diagnosis. And then about 2 weeks ago, a paper came out that talked about the brain being washed at night like a dishwasher and the cells getting small, and that the amyloid plaques were washed away with the other toxins. I'd just like you to comment on all that.
I was hoping you could take the trash can analogy to the dishwasher. Well, we do know that sleep apnea can cause memory problems by itself, so we do encourage people to get tested for it and treated for it. We also know that sleep apnea puts people at huge risks of a lot of blood vessel disease in the brain as well, heart attacks, strokes. So it makes sense that we'd want to treat that to decrease those risks. And I know there have been data on sleep cycles and the amount of sleep and amyloid. Maybe you guys can comment on that.
I'm read that study to a degree. I haven't gone through it with a fine-tooth comb, but it's an association study which suggested that people who had better sleep were less likely to be demented. And it's hard to know exactly where that comes from. As I think Bruce mentioned earlier, we know the pathology in the brain in Alzheimer's occurs 10 to 20 years before you develop symptoms. So is some of that association because Alzheimer's affects sleep cycles? I don't think anybody doubts that treating sleep apnea is a big benefit, that if you've already, even if it's not directly related to the Alzheimer's, it affects your memory. It's not surprising that when it's adequately treated for sleep apnea, he does better because he has only the Alzheimer's. He's not fighting the sleep apnea effect as well. So, but I don't know how you can't force people to sleep more than they, right? But I mean, one of the questions that's raised by a study like that is, should we be taking sleep medications, et cetera? We know generally that I like to avoid sleep meds if I can because they tend to fog cognition. So, and thinking skills, if anything.
But there's also data that's kind of interesting from mice, but where they can actually study though sort of the relationship between sleep and then some of these amyloid production and clearance. And actually there it looks quite interesting. There's a group at Washington University who's done really, again, some of these really amazing experiments where you can show that actually amyloid levels do change based on sleep patterns. And it's pretty compelling work. Again, how that relates to Alzheimer's disease I think is still another step. But it does suggest that somehow there's a linkage between sleep state and how much amyloid is produced. So, again, could there be a link then when you have sleep disturbance to then, that it could drive amyloid? It could be. But again, I think we'd have to actually study that per se to really determine if it's really driving disease as opposed to, again, as I think Dr. Leveren's suggesting, which is actually it's because of disease already occurring. So those are big questions.
There's a question up front here.
I was just curious, as researchers, is there still a need for human brains after someone passes that has been impacted by the disease? We used to I believe we used to have a brain bank in Cleveland that we no longer have, and just curious.
So his question is about whether there is a need for brain banking, and I think yes, there is, and I think there is still a small program actually at the university hospitals. Is that right, Alan?
That's true, yeah.
As we discussed just yesterday at a meeting in San Diego, and so I think we do feel that there's a need for that, and I think that's something we will be working on to actually enhance that program as time goes forward, especially as people who are doing the more basic science often come to me and say, we found a new protein we think is linked to the disease. We want to see what's going on in the real disease, in the mouse brain. Now let's take it to the human brain and see what the relationship is. So I do think the one thing that we've found in our brain banking in Seattle has been it's most useful if those individuals have also been studied extensively during life, so we can compare what we see in the brain after death to what was found along the way as they were going through the disease process. And as Brian mentioned, we also need non-Alzheimer people too, because we need to know what's going on in the normal brain. Alan?
Yeah, I think
Do you want to introduce yourself?
Alan Lerner. I'm from University Hospitals. I study Alzheimer's disease and treat it. So I think it's really important. This panel has highlighted the difference between things that reduce risk and things that are used in Alzheimer's disease, like statins. Epidemiology would suggest that statins, lower your risk, but treatment trials have been kind of a dud. Ditto estrogens, ditto non-steroidal anti-inflammatories, ditto many other things. So I think that these are really two different phenomena, which is a prevention trial versus a treatment model. And so when you read these in the newspapers, you really have to look at who is getting treated. Is this an aging thing? Is this a prevention thing? Or is this a treatment thing? So that's just a general caution for the audience.
As clinical investigators, what do you see as the barriers to enrolling patients to trials for Alzheimer's?
Number one is knowledge. Number two is getting an accurate diagnosis. I think those are just the major things. And having the studies, the money for the studies, obviously.
I think connecting patients and researchers together and each of them knowing about each other. So efforts to get out like this and educate the public about what's going on are really good ways to connect people so people know things are happening. I mean, there's quite a list at the university hospitals that are already going on here. We hope to expand the programs as well at Cleveland Clinic, and we need I think education and connecting people with the disease, their families, and with the researchers are the most important thing. Because I think generally people are interested in helping.
2 more questions.
Gentlemen, thank you very much for your time this afternoon. It's been very enlightening. I'm very interested about the inflammatory angle and want to know if there are any studies being done looking at other diseases, particularly autoimmune diseases such as rheumatoid arthritis, arthritis in general, psoriasis, and any link or any predisposition to Alzheimer's disease development.
You're the inflammatory guy. I can't remember if there's been anything. She did.
There's not huge amounts of data out there, actually. Looking at this, I'd sort of say in any really careful kind of way, sort of saying whether it could be or not. What's interesting, though, is that like this one gene that I mentioned, this TREM2, which was seen as a risk factor for Alzheimer's disease, it's now looks like it's a risk factor for frontal temporal dementia in addition. And potentially even Parkinson's disease. There's a couple of reports out on. So I think what that suggests to me is that there could be common mechanisms amongst a bunch of different neurodegenerative diseases, and inflammation may be one of those things. That, again, there's a normal change with aging in terms of the inflammatory pathways in the brain. And could this be, again, some way that the brain is either clearing debris from the brain, or is it ways that, again, are so responding to the damage that's occurring? And I think those are all good questions, but I don't know of any exact data specifically looking at unique populations. There is an autoimmune based dementia that can occur as well. So that's but that's completely different. That's not an amyloid driven, and in fact usually is treatable, I think, with steroids and can be treated quite effectively. But that's about all I know.
I did see a study recently in frontotemporal dementia where they saw an association between different types of FTD and people with certain kinds of autoimmune disorders. So I think this is something that people are looking at, and it may be, as we talk about, we don't all have the same kind of Alzheimer's disease, and these factors may play an important role in how we treat people.
Okay, last question.
I'm interested in knowing if there are any local studies being done on adult children of current Alzheimer's patients. That's for any of
There
you.
is one study called the Diane study, which is for a certain form of Alzheimer's that's caused by a genetic mutation. So usually about 50% of the family is affected. That tends to happen in younger people. And there's a big, actually international consortium that's looking at children of those family members. Not aware of people that are looking at children of sporadic Alzheimer's, are you?
Not that I'm aware of. No.
Okay. So thanks for coming, and I'll turn it over to Nancy.
I want to thank our amazing panel tonight. Every time I hear you gentlemen speak, I learn more and more, and eventually I'm going to actually be able to repeat that to somebody. But I think everybody would agree that it's a fascinating field. It's an important field. I can't think of anything else, quite honestly, that's more important right now. And we all thank you so much for the work that you're doing and that you're dedicated to. I want to remind everybody, again, Tricia told me I had to do this, of the materials you received. Tricia's in the back. Tricia, will you raise your hand? Tricia Vidovic of our staff is responsible for everything working perfectly tonight. Thank you, Tricia. Please complete the evaluation form. It's really important. This helps us in our planning as we go forward for next year's Foley. If you need continuing education credit, please go to the registration table outside, and you have to take your evaluation with you to get the certificate. And, hold on a minute, there are two things that you can do tonight to contribute to leading-edge Alzheimer's disease research. The first is to take part in the Alzheimer's Association's Trial Match service. Scientists have been breaking ground on new clinical trials, but one of the obstacles that has been reported is finding healthy volunteers to participate. The information you received on your seat, which is a postcard, will allow you to sign up for Trial Match. Please take a few moments to complete the card before you leave, and our trusty staff will be all over the place outside to collect the cards as you leave. The second action that you can take before you leave tonight is also extremely important, and that is to join our efforts and become an Alzheimer's advocate. And our public policy chair, Gail Sands, is over there. If you would wave, Gail, or even stand up for a second. Any of you who are interested in joining our public policy committee, we always are looking for new members. Alzheimer's disease is currently the 6th leading cause of death in the country. The first ever National Alzheimer's Plan, initially released in May 2012, was mandated by the Bipartisan National Alzheimer's Project Act, which Congress passed unanimously in 2010. Can you think of anything else that Congress has passed, let alone unanimously? This was extremely, extremely exciting for those of us in the Alzheimer's world. A new update was released this June with a new timeline for achieving its first goal, which is to prevent and effectively treat Alzheimer's disease by 2025. So that this plan called for by Congress can be translated into results, Congress must continue its leadership by supporting the necessary funding levels for the fight against Alzheimer's. We can't afford to wait. We need to take action now. Please sign up to be an advocate and join us in telling Congress that we can't wait any longer for a treatment or a cure. that the time to fund research at a level that will really make a difference is now. And I'm going to ask you one last thing to do, which is to help us raise awareness of the Alzheimer's Association's free programs and services. Tomorrow morning, please tell at least 5 people where you were today. And think of the magnifier effect if all of you get out and do that. I want to thank you all for joining us and your continued support of the Alzheimer's Association Cleveland Area Chapter. If you have any questions about the free services provided by our chapter, you can stop by our information table outside. I also want to thank Bob Roth for being with us tonight, who generously supported and sponsored this program today. Thank you, Bob. And good night to everybody. Thank you.